اوكي تمام. ازيكم الاول؟ I hope يعني الاجازه is treating you well. احنا اتفقنا هنتكلم مع بعض. اوكي anyways طيب معلش استاذنكم again، احنا هنتكلم مع بعض السيشن دي، فبليز كلنا نعمل unmute.
تمام.
خلينا نبدا السيشن بتاعتنا. طيب حد كده يقدر يفكرني احنا السيشن اللي فاتت اتكلمنا عن ايه؟ بدأنا نتكلم عن توبيك اللي هو كان البيولوجيكال موليكيولز. ممكن حد يفكرني بالظبط خدنا كام نوع من انواع البيولوجيكال موليكيولز، واحنا عندنا اصلا how many biological molecules do we have؟ No. Anyone can answer, please? Okay, just raise your voice, please, Yasmin.
We took about the lipids and the carbohydrates.
Yes, great.
And
well,
I believe we still have the proteins and the nucleic acid.
yes, great, excellent, Yasmin. So, exactly as Yasmin said, المرة اللي فاتت احنا اتكلمنا عن 3 kinds of the biological molecules. So we started with water and carbohydrates and lipids. And today, inshallah, we're going to continue with proteins and a very important part, which is practical testing for each of these biological molecules. And finally, we're going to end our session with DNA. So for today's session, inshallah, our lesson be for proteins, practical testing, and finally DNA. So before I start today's session, I just want you to interact and remind me of some questions about the last session. So here I ask you, so I have around 9 questions, so I want each student for 1 question, okay? So I'll start with, I think, Ahmed. Can you tell me, Ahmed, what are biological molecules? Just from what you remember, I don't need an exact definition. Yes.
Okay. How the exact definition?
No. Not
Can
an
I say an
exact
example?
definition. Yes, of course.
Yeah. اسمه ده. Yeah, like carbohydrates, lipids, what the stuff that we took اسمه ده previously يعني.
Okay. And what is the importance of biological molecule? They enter in what? So if we talk about our bodies, for example, they are composing, they are the structure of our bodies and of every biological organism. Okay? Okay. Just wait for one more question for you, Ahmed. Water is biologically important in? I remember that you answered this question last time.
Transportation and
Yes,
all around the body واسمه ده.
Yes, great. And this is due to the water ability to?
Due to the what, sorry?
Due to the water ability to do what?
Water's ability to do what?
It's a great. So I give you an option. It's a great solvent or solute.
It's a solvent. Okay.
Okay, so it's a great solvent. So
okay, now I understand. Okay.
It's a great vehicle for transportation of substance all over the body. Okay, excellent, Ahmed. Thank you. So Shahd, I think there is Shahd here.
Yes.
What is the main? Yes. What is the main? Okay. What? You're new.
لا أنا assistant أصلا مش student.
Okay. Okay. Thank you. Sorry.
عادي بس.
Okay. So Yasmin.
Yes.
Can you remind me of what is the main function of carbohydrates?
For providing energy.
Yes, great. So, and can you remember what is the chemical formula of carbohydrates? Yasmin?
CHO, and it depends on the kind of carbohydrate, I believe.
Okay. بس في حاجة. دلوقتي if I ask you the chemical elements of carbohydrates, you will tell me carbon, hydrogen, and oxygen. Okay. But the general formula of carbohydrate is something else. Can you remember?
I believe it's C and then H2 and then O.
Okay, CN, H2N.
Oh yeah, CN, H2N, O.
Okay,
N.
great. So, anyone else who needs to be asked? Classify carbohydrates. في حد يحب يجاوب ولا اختار أنا؟ Anyone else other than ياسمين وأحمد؟ Okay, طيب ياسمين, can you classify carbohydrates since إن أنت اللي بدأتي مع الcarbohydrates?
Okay. There are 3 types.
Okay, great.
Monosaccharide, disaccharide, and polysaccharide.
Actually studied this session, I think. Okay, regarding the polysaccharides, what do you know about the polysaccharides?
They are made of many simple or many monosaccharides, which is mainly glucose that we took.
Okay, so
And
like
there are example like starch, glycogen, and cellulose.
great, excellent, Yasmin. So back to Ahmed or anyone else who wants to be asked. Okay, can you give me an example, Ahmed, of a monosaccharide?
A monosaccharide. Honey?
No,
Wasn't
never.
it? No?
I'm talking about a monosaccharide. So it is something inside the honey. You can never say a
Glucose?
honey is a sugar. Yes, excellent. And what else?
Glucose and
Something else in the honey as well.
sucrose?
Nope. Sucrose is not a monosaccharide, actually.
Okay, so it was glucose.
And something
And
that starts with F.
with what? With what?
F, so fructose.
Fructose, okay.
Yes. Okay. Okay, Doha, we have Doha here. Chemically, Doha, lipids are made of. So I'm speaking here about the chemical elements that are making lipids, Doha. Doha, where are you? Can you unmute yourself, please? So you can answer in the chat if you can. Okay, I'll help you. So I'll choose Yasmin again. Chemically, Yasmin, can you remind me of the chemical elements of lipids? They are exactly the
Carbon,
same
hydrogen,
as.
and oxygen.
Okay, this is chemically, and Ahmed or Doha, if you're here. The elements of lipids, they join together to form two kinds of molecules. Can you remind me of these two molecules?
3 fatty acids.
Yes,
with
great. So this is one kind. And one glycerol. And glycerol is a kind of what? Can you remember?
Glycerol is a kind of what?
It's a kind of.
Is it a polysaccharide?
Nope, it's an alcohol.
It's an alcohol. Okay, my bad.
Yeah. Okay, so this is a question for both of you, Yasmin and Ahmed. There are two similarities between lipids and polysaccharides. Can anyone let me know what are they?
I believe they're both for providing energy.
Okay, great. And Ahmed, another one? Another similarity?
Another similarity.
Mm-hmm.
Maybe they're insoluble, both of them.
Okay, yes, great, excellent, Ahmed. They are water insoluble, both of them are water insoluble. So, can you mention a lipid function related to climate? Hey. So, Yasmin?
Lipid function related to climate?
Yes.
Yeah, it could have to do if the weather is cold, fats are burned down to warm up the body or
Before they are burned out.
something like this,
Before,
maybe?
adipose tissue, which is a layer just beneath the skin that forms an insulating layer from the very cold weather. right? Can you remember? Like polar bears, some animals in the North Pole. Can you remember such a thing?
Yes, yes. Sorry. The layers of fat are used to warm up the body.
Okay, so just stay with me. Carbohydrate is stored in plant as,
Starch.
yes, great. And in animal as?
Glycogen.
Yes, great. So كده انا طمنت عليكم بالنسبه للسيشن اللي فاتت. Thank you جدا for sharing. دلوقتي بقى نقدر ان شاء الله نبدا today's session. هنتكلم زي ما اتفقنا عن البروتينز، practical testing مش بس البروتينز والكربوهيدراتس and lipids، وكمان في الاخر هنتكلم عن الدي ان اي. تعالوا كده نبدا نتكلم عن البروتينز. So chemically speaking, proteins they have a similar structure to carbohydrates and lipids, but I cannot say the same. So similarity like what can you expect? So lipids and carbohydrates both are formed from carbon, hydrogen, and oxygen. Protein is similar, شبههم شوية. ففعلا هو هنلاقي عنده الكربون، الهيدروجين, and oxygen. But what makes a protein is another very, very important element, which is N. ولو تلاحظوا, protein is the only one that has N here, so it has the N here. Okay? Lipids and carbohydrates didn't have this N. يبقى لحد هنا البروتين شبه الكربوهيدرات and الليبيدز. لكن في essential element لازم يكون موجود في الموليكيول اللي معايا عشان ابدا اشوف اصلا هو بروتين ولا لا. وكمان sometimes ده مش essential element قوي، بس كتير من البروتينز بيكون فيهم sulfur. وبالتالي لو انا جيت سالتك the chemical structure of proteins هتقول لي ايه؟ chains. يبقى هل البروتين شبه الليبيدز والكربوهيدرات؟ Of course yes. Actually, very similar chemical structure because it contains carbon, hydrogen, and oxygen. However, it must have a nitrogen element as well. Sometimes it may have sulfur. Okay? Okay? Hey. Please unmute yourself. Okay. So, I have a question here for you. If I ask you, list only 4 chemical elements that are found in proteins, تفتكروا the answer should have what? I have only 4 options, so I cannot state the 5 elements. So the 4 options, priority would be for مين؟
Carbon, hydrogen, oxygen, nitrogen.
And nitrogen, exactly. ما ينفعش يبقى هو مديني 3 اماكن اروح انا كاتب له CHO and S. انت لو ما كتبتش الام مش هينفع تاخد الدرجه اصلا. بروتين يبقى النيتروجين ومعاه الكربون والهيدروجين والاكسجين sometimes sulfur. اوكي؟ واضحه البوينت دي؟
Yes.
Okay, great. So protein molecules يا شباب 3D speaking, chemically speaking they are chains. 3D wise they are made of long chains of monomers or small molecules. The small molecules that are making the protein are called amino acids. So, from your background, what do you know about an amino acid? Or at least from its name, it must have what?
Acids.
So, it must have an acidic function group, okay? Which is the carboxylic group. Great, excellent. And from the word amino, it must have what?
Protein.
Nope. Proteins, actually, amino acids are the component of a protein. So if you have a large protein, like a piece of meat, for example, and you digest this meat to its very, very fine components, it will be amino acids. Okay? The amino acids are joined together by certain kinds of bonds or chemical bonds in order to form such small chains or such long chains, sorry. Okay, so we have the amino acids, which are small molecules, that are joined together to form a long chains. Again, I'm reminding you, carbohydrates, lipids, proteins, and DNA all are polymers. So if you have a polymer, a large component or a large compound, they must be composed of something smaller. So in our case here, in the protein, it is the amino acid. Okay? Okay. Let's see how is the amino acid formed or what is it composed of. The amino acid structure is very important. As you just said, I have an amino acid, so it must have an amino group, like here, which is NH2. Okay, this is called an amino group. And as well, it has the acidic group, which is the carboxylic acid group. Okay, and it must have a central carbon and a hydrogen atom. So for an amino acid to be so, it must have 3 or 4 features of the structure. A central carbon attached to a hydrogen atom, an amino group, and a carboxylic group. Again, a central carbon attached to a hydrogen atom, an amino group, and a carboxylic group. Okay, is it clear? Clear for you?
Yes.
Okay, so we have Doha here. Okay, she's not unmuting herself. Okay, anyone else? I just want an interaction.
I'm here يعني.
Okay. So is it clear for you?
Yes.
So if you just make the slide disappear and I ask you, an amino acid is formed of, or it must have 3 features. What are they?
A central
Yes.
carbon
Great.
and an amino group and a carboxylic
Yes,
group.
excellent, Yasmin. Okay. We have around 20 different kind of amino acids. Okay. بس الفكرة في ايه؟ The arrangement of these amino acids. ال 20 amino acids دول ممكن يترتبوا زي التشين كده بالظبط. ممكن ياخدوا شكل التشين بس او زي شكل السبحة. كل مرة انت بتغير ولو single molecule بتعمل له shifting from a position to the other بيتغير معاه شكل السبحة مثلا كلها او شكل التشين بتاعتي كلها. فبالتالي لو انا عندي هنفترض انه ده كده بروتين هنسميه بروتين X مثلا. البروتين X ده is composed of 3 amino acids, the red one, the yellow one, and the blue one. In this very order. يعني ما ينفعش الأحمر يجي مكان الأصفر ولا الأصفر يجي مكان الأزرق. هما ماشيين كده من الشمال لليمين red, yellow, and blue. أوكي؟ لو حصل أي shifting بالشكل ده اتغير الارينجمنت بتاع الامينو اسيدز في التشينز دي هيبدا يحصل ايه يا شباب؟ هيديني بروتين مختلف تماما a totally different protein. وبالتالي انا اقدر اكونكلود من هنا ايه؟ انه الاوردر بتاع الامينو اسيد is very important for the protein shape and function. Again يبقى الاوردر بتاع الامينو اسيدز in the protein it must be in a precise order in order to give me a certain protein with a certain function. لو حصل أي tiny change في الارينجمنت ده بيديني بروتين مختلف تماما بفانكشن مختلفة تماما. النقطة دي واضحة؟ النقطة دي مهمة جدا. أوكي؟ يبقى أوكي تمام جدا. طيب أنا عايزة بقى أسمع منكم أنتوا الفانكشنز بتاعت البروتين اللي تعرفوها في الحياة بتاعتنا. البروتينز مهمة في إيه؟
one of the functions that I know, I think they are strands in the DNA. صح؟ البروتين ولا amino acids؟
Amino acids, okay. Something that's very popular nowadays, which is
growth in muscles يعني ولا؟
Yes, بالظبط. Exactly. أنت مش بتشوف دايما الولاد وهم رايحين الجيم. جيم يعني بروتين. لو عايز يعمل building للمسلز، المسلز بتاعتنا كلها
Yes,
دي
yes, I know.
معظمها
I know.
المين ستراكشر بتاعها عباره عن بروتين. تلاقيهم بياخدوا بروتين شيكس قبل ما ينزلوا الجيم. ليه؟ علشان المسلز، والمسلز هنا هي اللي ليها دور في السبورت بتاع البادي كله. اوكي، طيب ايه كمان؟ بروتينز داخله في ايه كمان in our bodies؟
and the hemoglobin and the for the oxygen
ضحى. اوكي
carrying.
sorry ياسمين. ضحى is raising a hand. اوكي ضحى you're unmuted. Okay.
It make hormones and enzymes.
Okay, it make hormones and what?
Enzymes.
Enzymes. Okay, great. Thank you, Doha, for contribution. So, as Doha just said, and I'm coming back to you, Yasmin, the proteins are forming the enzymes and hormones in our bodies. What about the enzymes? They are important in what? Yasmin, it's your turn.
And the chemical reactions around the body.
Yes, great. So, for example, if you eat a meal, you have the digestive enzyme that they will digest this kind of meal, this kind of food, and it will give you the fine components or the fine nutrients to all around the body. Okay, great. What else? What do you think the proteins function even in our daily life? Where can I find protein the most in food? Foods that are the richest in proteins.
Meat.
Yes, great, excellent. And what else?
Dairy products sometimes, I think.
Yes, great. Dairy products like cheese and so. So this was just like a mind of brainstorming for the functions of protein from our daily life. Here I'm talking about the function of proteins in another bit scientific way. So protein, as I said, they are the main component of muscles. Not only muscles, they are the main components of our cells. Of course, after water. So inside the cell, actually, the cell membrane itself and the organelles inside the cell all are made of basically proteins. Actually, if you have a wound, لو حد اتعور، التعويرة دي مش بعد شوية بتكون like something like قشرة كده تبقى ناشفة شوية، بعدها بيحصل ايه؟ just beneath اللاير دي بيبدأ يحصل generation للنيو cells. عشان كده مهم جدا لو حد wounded after an accident or so, a major accident، ان هو يبدا يدخل بروتين كتير في الدايت بتاعه. او مثلا البيرن بيشنت، لو حد حصل له لا قدر الله بيرن كبير، درجه كبيره من الحرق، لازم يدخل في الدايت بتاعه بروتين كتير، عشان ده مهم جدا للجنريشن بتاع النيو سيلز، علشان يحصل جروث وريبير للwounded او الانجريوس بارت او الدامجد بارت. طيب مهم في ايه كمان الفانكشن؟ ايه تاني بروتين بيدخل فيه في جسمنا which is very important؟ طبعا كلنا سمعنا الفتره بتاعت كورونا عن كلمه انتي بودي كتير جدا جدا. اوكي، ايه هو الانتي بودي؟ الانتي بودي يا شباب هو المسؤول، هو it's like an immune weapon، مسؤول انه يعمل like neutralization، يمسك ويقفش كده في الحاجه الانجيريوس اللي داخله لنا لجسمنا like the virus, the bacteria or so. الانتي بودي composition بتاعه mainly protein، اللي هو الحاجه اللي بتكون Y shaped like this. هناخد ان شاء الله الستراكشر بتاعه in detail بعد كده. بس مهم قوي عندي ان انت تبقى عارف ان البروتين ليها دور كبير في الاميونيتي بسبب الانتي بوديز. طيب في نقطه بينا او هنت كده هنا محتاجين نعرفه. دلوقتي يا ترى تتخيلوا كده الانتي بودي بتاع فيروس كورونا هو نفسه الانتي بودي اللي بيطلع مثلا against hepatitis C او HCV؟ حد كده يقدر يقول لي من اللي احنا لسه قايلينه just 2 minutes ago؟ هل يا ترى
No, they're different.
So again, I'm repeating my question. The antibody for coronavirus, is it the same as the antibody for hepatitis C virus, another virus? Someone
I
has
don't
said
think
no.
it's the same. No.
Yes, exactly. It's never the same. Why? because antibodies are very specific. They are specific for a certain agent, certain injurious agent, certain bacterium, certain virus. Every virus and every bacteria has its own antibodies. طيب هل يا ترى ينفع انا بقى دلوقتي لو انا عندي الانتي بودي ستراكشر مثلا بتاع الاتش سي في، هل ينفع اخده زي ما هو كده واشتغل بيه على الكورونا؟ اكيد لا. طب ايه اللي حصل علشان يبقى هما الاثنين انتي بودي؟ يعني ايه اللي حصل علشان خاطر ده يشتغل على الكورونا وده يشتغل على الاتش سي في؟ تفتكروا ايه؟ ايه اللي اختلف في الاستراكشر؟
maybe the arrangement.
Yes, exactly. Excellent. Great. The order of the amino acids. For example, لو أنا عندي في الانتي بودي بتاع كورونا فيروس كان الارينجمنت A B C D X. في ال HCV هيحصل الآتي A C D X or something another amino acid or the arrangement will differ. Just the arrangement. Okay? فمهم جدا ابقى عارف ان الاوردر بتاع الامينو اسيد في البروتين تشين مهم قوي للفانكشن وللشيب ككل بتاع البروتين. Another function of the protein زي ما اتفقنا ان هي the structure of the enzymes، والenzymes يعني metabolic reactions in all of our bodies. ايه كمان؟ دي حاجه بقى ما نقدرش نقول عليها انها main function للبروتين، بس هي موجوده وبتؤديها. تخيلوا معايا حد كده تاه في الصحراء لمده شهر، وتعالوا نطبق اللي احنا خدناه السيشن اللي فاتت ونربطه بالسيشن دي. هو تاه في الصحراء، مفيش اي ريسورس للميه للاكل، مفيش اي حاجه داخلاله الجسم، فهيبدا البادي in order to survive ان هو يعمل utilization للكربوهيدرات اللي في جسمه، صح؟ like what؟ stored as a carbohydrates بتكون stored in our bodies as a
glycogen.
حلو قوي. ففي اول كام يوم من التوهان بتاعه ده هيبدا البادي يوتيلايز الكربوهيدرات اللي stored as glycogen in the muscles and the liver. دول يا دوبك يكفوه يوم يومين بالكتير. بعدها بشويه هم طول، يبدا البادي يلجا لمين؟ مين السكند بعد كده؟ البادي لازم هيلجا له as a source of energy لما يخلص على كل الكربوهيدرات. when it runs out of carbohydrates مين السكند source of energy؟ خدناه السيشن اللي فاتت. lipids. اوكي؟ طيب لو هو بقى خلص الليبيدز كلها، خلاص الراجل ده رفع جدا، خس تماما في الشهر ده، هيتبقى له مين in order to survive؟ ما قدامهوش حل غير المسلز ان هو يبدا يعمل بريك داون للبروتينز اللي في المسل ويحولها somehow into glucose علشان تبقى a source of energy as well. وبالتالي اللي انا عايزه اقوله هنا ايه؟ انه one of the functions of the protein is it's an energy source. However, it's not the main function of protein. بالعكس، احنا عمرنا لو حد طبيعي في الستيت الطبيعيه البادي will never use protein as an energy source. Is it clear point دي يا شباب؟ لو
teacher.
اوكي سامعيني كويس؟ فاهمينها تمام؟
yes.
Yes.
اوكي حلو جدا. طيب دلوقتي حد يقدر يفكرني كده باكزامبلز لبروتينز في حياتنا بقى اليوميه، بروتينز ملموسه نقدر نشوفها نتعامل معاها بناكلها بنستخدمها؟ Hey, examples of proteins. Proteins, examples of proteins في حياتنا اليومية. In our daily
meat
life.
يعني
Yes, حلو قوي. ايه كمان؟
dairy products cheese
اوكي، ايه تاني؟
Our nails, like they're
Great,
made, I think, of
great.
protein.
Yes, our nails, the keratin protein. Excellent. Else, what else? In our blood.
The hair too.
Okay, in our blood. A very important protein in our blood. What is it? The one that carries the iron.
The hemoglobin.
Yes, excellent. Hemoglobin is actually a protein. So here we have some examples of protein. we can classify protein into two classes. Soluble proteins, water soluble, and insoluble proteins. The water soluble proteins, for example, are like hemoglobin, which is the red pigment in our blood that carries the iron, which is very important for respiration. Okay? And the other type, which is the insoluble protein, so it's not like soluble in water. So we can imagine here our nails. Actually, our nails are made of protein, a protein which is called keratin. Okay? And our hair too, it's made of keratin. However, it's insoluble. Just عايزاكم تتخيلوا معايا if this keratin was or were soluble, ايه اللي كان هيحصل؟ Would we ever have nails? Would we ever have hair? No? Yes, excellent, Omar. Thank you. Can you unmute yourself, Omar, and join us? Okay. So, just as a recap of the protein we talked about. So today we started with the structure of the protein. The structure of the protein, chemically speaking, it's formed of chunks: carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. 3D-wise, a protein is formed of long chains of the small molecules, which are the amino acids. An amino acid must have three features, which are, can anyone help me? Omar, can you please help me if you're hearing me? Omar, Doha, any new one who wants to join us, or even in the chat? An amino acid structure. So Yasmin or Ahmed. Yasmin?
The central carbon,
Yes.
the amino group, and the carboxylic group.
And the carboxylic group. Thank you, thank you. So, and then we talked about that the order of the amino acid in the chain is very important for the shape and the function of the protein. And finally, we talked about both the functions and some examples from our life of the proteins. And we agreed that some proteins are water soluble, like hemoglobin. Others are insoluble, like keratin in our nails and hair. Okay, so I think now it would be time for a little quiz. So can you please, all of you, go to the book widgets in the classroom and start by solving quiz named Pop Quiz Trial Session. Pop Quiz Trial Session, this one. It's already posted. Pop Quiz Trial Session 2. So back to the session, شباب. I think you've all submitted the quiz now. اوكي يا شباب، خلصتوا الكويز؟ كلنا رجعنا؟
لا انا لسه مخلص بس هو the first question the first page هو مش باينه لي قوي اصلا. انا للاسف على الموبايل بس مش باينه لي يعني صح ولا لا.
Because it's a picture to be annotated. Probably علشان الموبايل. بس أنا ممكن يعني هي about the amino acid structure. فإن شاء الله لما تفتح من اللابتوب أكيد هتبان. ممكن دلوقتي أنا برضو أوريها لك. بس كلنا خلصنا كده يا شباب، يا ريت بقى نرجع. think more than enough. هنرجع تاني لأول سؤال ده بس after some time. عشان الجزء الجاي بقى ده مهم جدا جدا. ده مش للسيشن بتاعتنا النهارده بس. ده مهم جدا ان شاء الله يعني نكمل معانا في الاو ليفل كله في كل البراكتيكال اكسبيرمنتس اللي بنتكلم عنها. طبعا البيبر 6 هي alternative practical بتكون قائمه mainly على البراكتيكال اكسبيرمنتس. بالتالي مهم جدا ان انت بلاش ان احنا نقعد نمسك اكسبيرمنت نحس انها تقيله قوي مش فاهمينها علشان احنا مش شايفينها وانها كلها حفظ فبنحفظ الكلام زي ما هو والريزلت زي ما هي. احنا عايزين نخلي دلوقتي كده الموضوع بسيط جدا. في اي practical experiment ان شاء الله هتقابلنا على طول during الاو ليفل بتاعنا كله في البيولوجي. اوكي تمام كلنا هنا يا شباب؟ اوكي اوكي طيب دلوقتي احنا هنتكلم عن basics كده هنستخدمها ان شاء الله في اي practical experiment تقابلنا. دلوقتي يا شباب لو انا عندي practical experiment لازم ابقى بسأل نفسي 6 اسئله مهمين جدا عن البراكتيكال experiment اللي معايا دي. اوكي لانه اي experiment هنتكلم عنها او اي experiment عموما في الساينس بتكون يعني based على 6 elements. The first element is why am I doing this experiment? What is my purpose of doing this experiment? What's my target? وانا ليه بعمل كده؟ ايه الفاليو اللي انا بدور عليها from performing and conducting such an experiment. الحاجة التانية المهمة جدا الاليمنت التاني what am I searching for? What's my target? انا بدور على ايه هنا؟ يعني why هي الفاليو انا ليه بعملها. لكن what انا بدور على ايه؟ حاجة تحديدا chemically speaking او biologically speaking التارجت بتاعي ايه؟ حاجة very specific. يبقى الواي غالبا بتكون broad شويه، الوات غالبا بتكون حاجه very specific. والهاو ايه هي الستبس بتاعت الاكسبيرمنت، هعمل الاكسبيرمنت دي ازاي. والموضوع لو احنا فاهمين why and what يبقى سهل جدا نفهم how to do such an experiment. بعد كده بيجي دور الobservation. يعني ايه بقى الobservation؟ the change of the result or the change of everything that's happening along the course of the experiment. يعني انت say starting with certain kind of powder which is dry. At the end of the experiment or during the experiment, it will turn into, for example, certain kind of liquid. This is an imaginary situation, of course. But my observation here is that a powder has converted into a liquid. Okay? So any change that's happening along the experiment, this is called observation. The final observation or the observation that has the most meaning is called the result. which is the positive and the negative of the experiment. Finally, وهنا بيقع كتير من الستودنتس. ليه؟ لأن أنت في paper 6 أنت مش بس ممكن يجي يسألك على الريزلت، وممكن جدا يسألك حواليها ويربطها لك مثلا توبيك هنا بتوبيك هنا ويربط لك الاثنين ببعض. تبقى المشكلة فين؟ إن أنت عارف الريزلت كويس أو حافظها كويس جدا. So you cannot interpret or explain what's behind this result. أنا ما يهمنيش إن أنا أبقى مثلا حافظ ألوان. Okay, by a test it gives me purple color. لا، مش ده خالص الهدف من التجربة. ولو ذاكرت بالطريقة دي الموضوع هيبقى صعب عليك جدا وهتبقى الاكسبيرمنت تقيلة جدا. المهم إن يبقى فاهم هو أصلا الكالر ده. طب هو هنا مثلا لو حصل الكونسنتريشن قال هل الكالر هيتغير؟ طب لو زاد الكالر هيزيد؟ طب هو انا لو ربطت بطابق تاني يا ترى انا خدت بروتين هنا وخدت بروتين في طابق تاني somehow مثلا في الانزيمز هل ممكن يجي يربطهم لي ببعض؟ طبعا. وبالتالي ان انت تعمل interpretation للداتا وللريزلت بتاعتك ده مهم جدا جدا as an element of الفهم بتاعك للاكسبيرمنت واستيعابك ليه. لو انتوا مشيتوا بالطريقه دي مش هتحتاجوا تحفظوا. اسال نفسك الست اسئله دول. وعلى فكره sometimes you will not find like an exact answer. هتحس انه اصلا ما فيش observation، هو كله خلاص اداني result على طول. sometimes هتلاقي ان ال why and what متشابهين and very similar. بس المهم ان انا وانا ماسك الاكسبيرمنت ما بقاش جاست بشوف انا بحط ده على ده علشان خاطر في الاخر يديني الكالر الفلاني. انا مش بحب الطريقه دي ابدا، ومش دي الطريقه اللي تخليك تبقى مطمن وانت confident وانت بتحل في paper 6. يبقى again تسال الاسئله دي لنفسك. يبقى انا بدور على why او البيربس، what ايه التارجت بتاعي، الستبس بتاعتي او الهاو تو، الاوبزرفيشن او التشينجز اللي بتحصل، result، واخر حاجه الانتربريتيشن او معنى الريزلت اللي انا وصلت لها او الاكسبلانيشن بتاعها هي ليه حصلت. طيب تعالوا نطبق الكلام ده على الثلاثه ايتمز اللي احنا خدناهم في البيولوجيكال موليكيولز اللي هم الكربوهيدراتس، البروتينز، والفاتس. لو انا حبيت اعمل تيستنج ليهم هم الثلاثه، تعالوا كده نبدا باللي احنا لسه مخلصينهم، او تعالوا نتكلم الاول عن هو انا اصلا التارجت بقى من كل التستنج اللي انا هعمله هنا. عارفين ده مشهور قوي. ده ايه؟ ده النيوتريشنال ليبل. اي برودكت انت ممكن تجيبه زي الشيبسي، التونه، الباتر، any food products it must have such a nutritional label. important جدا. طيب what are you finding here؟ هنلاقي انه مثلا مكتوب exactly there are 5 grams of sugar, there are total fat of كذا, and finally proteins of 5 grams for example. So a nutritional label ده احنا بنوصل له ازاي؟ يعني ازاي عرفنا انه for example the pack of tuna will have around 5 grams of protein؟ من خلال التستنج بتاعه البروتين ده. ان انا بدور على البرسنتج اوف بروتين، او انا مثلا عايز اعرف هو ده بروتين اصلا، البرودكت اللي معايا ده فيه بروتين اصلا ولا لا؟ yes or no. كذلك هنا for example in this chips bag انا عايز اعرف ال amount of carbohydrates، هو في carbohydrates مثلا ولا ده برودكت تاني؟ يعني حد اداني برودكت جديد خالص ما اعرفش الكومبوزيشن بتاعه ايه. هعرف منين؟ بان انا ادور على الكاربس، البروتينز، والفاتس اللي موجود. اوكي تمام لحد كده؟ فاهمين يا شباب النقطة دي؟ Okay. So let's start with proteins. There is a very important and very popular test which is called the Biret test for proteins. Simply, Biret test, all what you're gonna do is just you will mix your food with water, a sample of your food, for example, certain like milk or some kind of yogurt or just a sample of like meat or like chicken, but it must be very, very like tiny particles. And to mix it very well with water to make like an extraction of the amino acids and the proteins in there into this water. And then you add what's called as Biret reagent. Then you will just wait for around just a few seconds. If there is a protein in your sample, the color will turn from the blue color into the positive purple color. Okay? If there is no protein, the negative result will be that the color will stay as blue, as exactly the color of the reagent. And in order to imagine it a little bit better, I'm having a video here for you. So just please. So
the protein test in the reagent Bio-Ret to indicate the presence of peptide bonds in a solution. The first test tube contains only distilled water. Notice the Bio-Ret does not change color and remains blue when the solution is negative for proteins. The second test tube contains an albumin solution. Notice the Bio-Ret changes to a purple color, which indicates the solution is positive for proteins. The third test tube contains a sample of digested albumin. Notice the Bio-Ret changes to a subtle pinkish color, which indicates the solution is positive for amino acids.
okay. So as you've just seen here, if I have a sample of, for example, an egg, and this egg, of course, has an albumin inside it, which is like albumin in Arabic, it's the white of the egg, the white part of the egg, okay? So it's basically protein. How can I detect? So if you have the Bio-Ret reagent and you have this egg, you can simply extract a certain part or a few milliliters of this white part or the albumin of the egg, and you add the Bio-Ret reagent, just a few drops, and the color will turn from blue to purple or mauve-like color. Okay? Okay. So here I'm having a question for you. So I'll choose Doha. Can you join us here? Even in the chat if you want to. Doha. And Omar. And actually, and Ru'a, I think. So here I'm having a question. هو بيقول لي ايه في الكويستشن ده؟ بيقول لي هو مديني الصورة دي بتاعت one egg. الايج ده الستراكشر بتاعها عبارة عن ايه؟ هي عندنا الايج شيل من بره وجواها two parts معروفين قوي. الوايت part which is called the albumin and the yellow part which is called the yolk. Okay? So okay, no problem. Okay, you can join us, Omar, just in the chat as you're doing. Okay, so here you have this egg, and the albumin is mainly composed of proteins. And he's asking you, a student has tested some albumin for the presence of protein using Bio-Ret reagents, and the solution has changed color. It was a positive result, of course, because the albumin is very rich in protein. What will the color change? So, Omar, you can just answer in the chat, or Doha, or anyone. Ru'a, I think. It turns to purple color. Yes, okay, great, excellent, Doha. Thank you. Thank you. So, before I move to that carbohydrates thing, here, if I want to apply the 6 elements or the 6 principles that while I'm studying an experiment, what is the value here? What is the value from just testing for proteins? And this is just for discussion. So, anyone? Why am I doing this? Why would I want to test for proteins? So, Yasmin?
To see if it's present or there.
Okay, so to see it's okay, it's present. Okay. Can you elaborate? So I tell you, for example, دلوقتي لو انا مثلا بشتغل في مكان ده like جيم مثلا، اوكي، انا لازم اعمل like a brochure كده. البروشور ده بيقول الاماونت اوف بروتين اللي موجوده في كل فود، او مثلا الفودز اللي very rich in protein والفودز that are not، عشان لو حد is going on a certain diet، لو حد ليه تارجت معين في الدايت بتاعه لازم يبقى عارف على الفود بتاعه ده contain certain amount of protein مثلا او لا خالص. تمام. كمان بيفيدني جدا ان انا اقدر اصلا اعمل نيوتريشنال ليبل اللي بيكون موجود على الفودز اللي احنا بنشتريها سواء في السوبر ماركت او من اي حته. تمام. ده كده الواي بتاعتنا. طيب وات انا هنا بدور على ايه؟ انا هنا بدور على البوند اللي بتعمل جويننج للامينو اسيد. طيب how؟ It's very simple. From the sample of the protein الفود بتاعنا and you add up a few drops of البايرت reagent. So the negative result will be, so if I have a control with water, the negative result will be hey, the negative result of Biret test is blue color or purple color?
Blue color.
Of course, yes. And the positive is, of course, the purple color. There is no observation here actually because just it's very, very fast reaction. Once you add the Biret reagent, it will change into the result, into the purple color if there is protein, or it will just stay the same if there is no. Okay? So the interpretation here. So for example, in this case of the egg yolk, the egg albumin, how can I interpret this in my own language? How can I explain the result of C? So I can simply just write down that. So in conclusion, the albumin contains a high amount of protein. Simply. Full stop. So the interpretation doesn't have to be a very complicated thing. No, it's actually very simple if you understand the aim of the experiment. Okay? Anyone has a problem with this?
Miss, I have a question.
Yes, of course.
How do we know if the concentration is high or low? Like it has protein, but if it's a high concentration or a low concentration of protein?
اوكي actually انت سبقتينا يا ياسمين. احنا كنا لسه هنتكلم ان شاء الله في الاكسبيرمنت الجايه عن الكونسبت بتاع quantitation or to know the quantity of something. However, just to answer you shortly here, this test is not quantitative. It is only qualitative, meaning it will only tell you if you have a protein or not. Okay? Sometimes it could be like, okay, the purple color will be darker when the percentage of protein is higher. However, you can never depend on this. There is lots of other methods, chemical methods, to know the exact amount of a protein in certain food, but it's not our scope here. Okay?
Okay,
You get
Miss, thank you.
Okay, great. So, regarding the carbohydrates testing, actually, we have two tests for carbohydrates. The first test is one for reducing sugar. So here, I want you to tell me now, the reducing sugar here is acts as which of the 6 elements? Is it the why, the what, or the how? What is it? So reducing sugars, if I'm saying that this is a test for reducing sugars, so the reducing sugar will be my target, right? Should be the something that I'm looking for. So it is the what of the experiment. Here, I'm trying to find the reducing sugar. And actually, what is the meaning of reducing sugar? Simply, chemically speaking, a reducing sugar is one that has a free ketonic group like CO or a free aldehyde group like CHO, like this one or this one. This is very simple. And it's called a reducing sugar. Why? Because it can donate electrons. However, this is not our scope here. All I want you to know is that this test is going to be for finding reducing sugars like, for example, glucose, fructose, and so on, the one that have a free ketone CO group or a free aldehyde CHO group. And this test is called Benedict's reaction. Benedict because it is for the reagent that we are using. So here we have, the test is very simple, or the experiment, the steps are very simple actually. You have the solution of your sugar or the sample of your sugar, and then you will start to heat it in a water bath around 100 degrees Celsius. After that, you will wait for some time, and then you're adding the Benedict's reagent. So here I have my solution, and just whenever it just gets a little bit hotter, I'll start to add my Benedict reagent, and I let it get to be heated for around 3 to 5 minutes. Okay? After that, I'll observe the color change. I'll observe that if I have a reduced in sugar in my solution or in my sample, the positive result will be as follows. The blue color of the Benedict reagent will disappear, and it will turn into green, yellow, orange, and a brick red. And this is my final result. And if it's totally negative, there is no reduced in sugar in the sample, it will just remain blue, exactly like the start. What about this spectrum of colors? I have here 4 colors, not only 1. This is why, actually, it's about here, and here to answer your question, Yasmin. It's about the amount of the sugar inside the solution. So, exactly like Yasmin has asked just a few minutes ago. Here I start with the blue. There is no sugar at all. However, if I have just a very, very few amount of sugar, or the reduced in sugar inside the solution, it will turn into green. If you have a higher amount, like for example 1.5 grams, it will turn into the yellow color. If you have more and more of the reduced in sugar, it will turn to orange color. And finally, the highest amount of the reduced in sugar, it will stay as brick red, or it will turn into brick red. Brick, actually, it is the color of brick. الطوب لو أحمر غامق جدا, brick red color. Okay? هنا الكونسبت ده مهم جدا نبقى عارفينه, which is it's called a semi-quantitative test. ليه بقى هنا؟ Yasmin, you can unmute yourself. ليه بقول على البنديكت تيست ان هو semi-quantitative هنا؟ Have you got it?
Because we can check the concentration of the amount present.
Okay, but actually it's not quantitative. It's still semi-quantitative. Why? Semi meaning half-half, like مش قوي, not very accurate. I can't know the precise amount of the sugar. However, I can have like a clue around the amount present in the solution. So this is the meaning of a semi-quantitative. Semi-quantitative means that the color or the result produced from the test represents or corresponds to a range of the amount, a certain range of the amount. But I can't say that this tube will have 0.5 gram. No. I know that it's around 0.5 grams to 1 gram, for example. So if I have the color right here, green, and I want to know the exact amount, Benedict reaction will not be enough. I have to perform another test on it in order to know the exact amount that is between 0.5 and 1 gram. Okay? Is it clear now?
Yes, miss.
Okay. So can anyone again recap this part for me? The Benedict test from start to end and his own interpretation of the test? Anyone? So Ahmed, if you're here. Hello. Okay, Doha, can you join? Yasmin, can you just recap the Benedict test for me?
Okay. So we add.
If I give
I'm
you a
sorry?
sample, of, for example, say grapes. I give you the
Great.
grapes. Okay, what are you gonna do?
So I'll crush the sample first,
Yes.
and then add it to water to make it a liquid or a solution,
Yes, to extract
and then add it to Benedict's solution and heat.
Okay, great.
And then depending on maybe the concentration, but not the exact concentration, maybe the
Yeah.
amount present, will get a color.
Yes, exactly.
And if there is no reducing sugar whatsoever, then it will be blue. Or
stay as
semi-liquid.
the original color of the reagent. Okay, excellent, Yasmin. Thank you. So here I have just a quick video again for the Benedict's test, just to see it in reality.
Wrappers, test solutions, Benedict solution, 100 degrees Celsius water bath, and hot mix.
So here, like, just see, here we
100
have
degrees
different
Celsius
solution
water bath.
of different extracts of that may be containing reducing sugar.
Label your test tube 1 through 8 and place in the test tube rack.
Just up to labeling your tubes, we'll start to add.
Here is a table of solutions we will be using. 1 water, 2 starch, 3 sucrose, 4 glucose, 5 apple juice, 6 coconut and orange juice, 8 soy milk. Add about a half a dropper of each solution to the appropriate test tube. Now add 10 drops of Benedict solution to each test tube. Place the entire test tube rack into the 100 degrees centigrade water bath for 3 minutes. Be careful, it's hot, so use hot mitts. After 3 minutes, pull the rack out of the water bath and set aside to cool for several minutes. Record your results. Clear blue indicates the absence of reducing sugars, or negative. Green yellow with precipitate indicates trace to low levels of reducing sugars, plus orange red with precipitate indicates moderate to high levels of reducing sugars, plus. Our results show that water, starch, sucrose, and soy milk give us negative results, whereas glucose, apple juice, coconut water and corn juice give us positive results.
Okay. So I hope you're all getting well along with this test. Okay. So how can we just apply the principle of the 6 elements to this experiment of Benedict's reaction? Why are we performing the Benedict's reaction? Why? What's my target here? What's my purpose? Hello? Okay. My target here is to detect the presence of, as we said, reducing sugars, the ones that have a free ketone group, CO, or a free aldehyde group, CHO. Okay. What are these exact reducing sugars? For example, we have the glucose. Okay, they have a CO right here. Fructose as well, CO. And galactose, for example, and much more examples. How? So we add the sample of the sugar to a Benedict's reagent. We heat it for around 2 to 3 minutes and we wait for the color to change. The observation will be multiple colors according to the quantity of the reducing sugar present. The merely negative result is a stained blue, and the extremely positive result, or the one with that's corresponding for the highest concentration of the sugar, is brick red. And here we can interpret this that the darker the color somehow, the higher the concentration of the sugar. Okay. So here I have a question for you from one of the past papers. So the question says that we have 2 types. In conclusion, actually, after you can read this. In conclusion, the question is saying that we have 2 kinds of pepper, okay? The red pepper and the green pepper. And the question claims that the red pepper contains more sugars than the green pepper. So it will taste like a little bit sweeter than the green pepper, which is actually a fact. So the question, all in all, is asking, describe how you could test if the sweetness of the red pepper is due to the presence of reducing sugars. So he's saying that actually it's a fact, the red pepper is sweeter than the green pepper, and just proves that this sweetness is due to reducing sugar, not any other sugar. What would be your answer? Simply, it will be the Benedict's test. So we will perform the Benedict's reaction on an extract of the red pepper, and you will see that there is a graduation in the color change, and this, if there is a change in the color from the blue of the reagent, then it has or it contains the reducing sugars. Okay, is it clear the way we solve it? Okay, so I have another one for you. That's your turn. Anyone who's interested to answer? Yasmin, can you join me here? Okay. Or anyone else? Actually, I don't know why are you not joining. Nru'a, Duha, anyone? Okay, just stay with me, Yasmin. So here I'm describing, this is a question, and you have a fruit, which is a strawberry. And he's asking you to describe how would you test the fruit to show the presence of the reducing sugar. It's a very simple answer, actually, and it's a very easy question and a very granted question in the exam. So can you help me?
Okay. So using the Benedict's test?
Okay. So how are you going to answer exactly like in the exam?
Okay.
Describe.
Firstly, question.
When he asks you with a describe, so you're going to elaborate a little bit. your answer.
Okay.
You cannot just
Uh,
answer with a single word. Okay.
Yes. Okay, so we crush and grind the sample or the strawberry, يعني. And then we
Okay.
add it to water to form a solution.
Mhm.
Then add to Benedict's solution and heat for a couple minutes.
Okay, great. So, to show the presence of reducing sugar, what would happen? If this is
And then
strawberry,
check for the color. It changed from blue to red, or maybe you can say also green and orange and yellow.
okay, excellent, Yasmin. Thank you. So, Yasmin, another question. Can you describe how would you test this fruit for the presence of proteins? Not
Okay,
carbohydrates.
so here we use the biology agent.
Yes, excellent. And it will turn to
Yes. So we crush and grind the food sample, add to water again. Then we add the biology agent. If changed from blue to purple, then it contains protein.
okay, great. Thank you. Excellent. So, the second test for carbohydrates will be the iodine test for starch. Actually, it's a very simple test that we all know about. I think we've already seen this many times along our learning journey. So, the carbohydrate test for starch. So here, all what we do is that we have a sample of the food. We add the iodine solution. What is the normal color of the iodine? The iodine's normal color is like orange-brown. If you add this orange-brown iodine to a sample of a certain food that's containing starch, like potato, for example, it will immediately turn into a blue-black color. Okay? If there is no starch in your food, then it will stay like in the orange-brown state. Okay? Like this. So here I have like a transaction of potato. Potato is very rich in starch. So this iodine is specific mainly for starch, not any other sugars. So if you just take this transaction of the potato and you add like very few drops of the iodine, immediately the iodine color will turn from the orange-brown color into the black-blue color. Okay? Okay. So, and here we have another question. Here he's given you like, actually this is just an accent of a certain question, the whole question, where the question is talking about certain carrot as a storage organ. So, knowing that a storage organ in plant will contain what? A storage organ in plant
Starch.
will contain starch, exactly. So, he doesn't have to mention this in the question. We already know it. However, is already mentioned, and he's asking you to test this carrot. Actually, it goes for the same for potato and sweet potato, sometimes onion as well. So he's asking you to test for the presence of starch in this fruit or in this kind of vegetable you have. So what are you gonna do? Again, you will add the Benedict agent and you will heat the sample. You will wait for 3 to 5 minutes and observe the color change from blue and gradually from the yellow, green, orange, and finally the brick red, according to the amount of sugar present. So, sorry, here I'm talking, sorry for this. Here I'm talking about the starch, not the reducing sugar. So here I will add just a transaction of this carrot. I'll add just a few drops of the starch, of the iodine, and it will turn from its original color, the orange-brown, to the blue-black color. Because the carrot is very rich in starch. So here we have like one more test in order to test for oils or fats and lipids. And actually, it's a very simple test. What is this test and how can we perform it? So here my target will be to detect the presence of fats in a certain sample. Sometimes we can see the oils, okay, and we know that this is a lipidic something. Sometimes it could be in a very low percentage that we cannot know just by naked eye. So what are we going to do here? So for example, you will take a piece of the butter, if this is your food sample, and you will chop it and crush it very well, and you will shake it up vigorously with ethanol. So here I'll have a test tube that's containing ethanol, and I'm gonna add my cube of butter. I will shake it very well, and then I will add some water. What exactly is gonna happen? Actually, let me show you this. The ethanol is a very good solvent for oils and lipids, whereas the oils cannot dissolve in water. So the ethanol will start to extract the oils from the butter cube or from the sample you're having, and it will start to like dissolve it a little bit in water. However, the droplets of these lipids or fats, food, it cannot dissolve completely in water. So it will form like very small droplet in a whole amount of water. And this is what we call emulsion. So if you go back to the test name is ethanol emulsion test. What is the role of the ethanol here? It's like extracting the fat from your food sample and trying to dissolve it into or homogenize it with the water. Okay? So here, if you add a piece of butter or whichever food that's containing large amounts of fat to a test tube that's containing ethanol, and you shake it very well, it will dissolve in the ethanol, and then you add water, it will finally give you a milky color solution or a milky color emulsion. So what is the emulsion? It is the liquid that's containing multiple droplets of fat or lipid substance that's not very dissolved in water. The most popular and the most common and most well-known emulsion is, of course, milk. Actually, the milk contains a huge amount or a huge percentage of water, and it contains cream and like fatty substances as well. So the dissolution of the fatty substance in this water to give you the final product of milk, this is called emulsion. So the milk is actually a kind of emulsion. Okay, is it clear? Okay, so here I have another question for you. Can anyone join and help me answer this question? Hello? So, Yasmin, can you join?
Yes.
Okay, so here I have an egg, okay? And as we agreed, an egg is formed from basically an egg albumin, which is the protein part, and the egg yolk, which is very rich in fat. Okay, the yellow part of the egg, which is the egg yolk, is very rich in fat. How can you test for this fatty part if it contains really any fat? How do you know the positive test, or how do you know the positive result of this fat present in the yolk?
We observe an emulsion when, do I say the whole or just the observation at the end? Like the
Nope.
whole.
Actually, you have to state here in this question, he is asking you to state what the student would observe because he's already carried out the emulsion test. So he's not asking you to state all the steps of the test, but I'm asking you to now. Okay.
So we add the food sample or the egg to ethanol, and we shake vigorously so that they mix together.
Mhm.
And then we add this to water.
Okay.
If fat is present, then a white emulsion or suspension is formed at the top of the water because they will not mix.
Mhm.
And if you see closely, there will be like tiny droplets of looking fat.
Okay, great. Thank you, Yasmin. Thank you. So now here we are for another quiz, just a very quick quiz this time, which is, well, it's called Pop Quiz, Pop Quiz 2, Trial Session 2, this one. So here we have pop quiz 2, trial session 2. Can you all join the quiz, please? We'll just start answering the question there. So we have around just 2 minutes to finish the quiz. 2 more minutes. So, seeing time's up. Can you please rejoin the presentation? So now, please can you rejoin the presentation? Okay. Here we're moving to the final item of the biological molecules. Now we're finished with water, carbohydrates, lipids, proteins. And actually, we're done with the testing of carbohydrates, proteins, and lipids as well. Now we're moving to the final item of the biological molecules, which is a very important one, which is DNA, or deoxyribonucleic acid. So here, just to start from the big picture, a human being is actually the structure of any human being is actually cells. Inside the cell is a nucleus. And inside this nucleus lies the DNA. In the form of what? In the form of chromosomes. So here, if we just pick one chromosome and start to detangle it like this, or just to spread it out, it will be the structure of the DNA. So if you start with the human being, you can just reduce it into the DNA. So again, human being, cells, inside the cell there is a nucleus. The nucleus contains the chromosomes. The chromosomes can be spread out into the DNA. Even smaller is a gene. What is a gene? A gene, simply, it's a segment of the DNA. Okay, so let's get just more familiar with DNA a little bit more. What's the DNA? The DNA, by definition, it's a chemical molecule that's responsible for transmitting the genetic material and the hereditary material from parents to offspring. So if you have a parent, if you have two parents, one is blonde and the other one is black, for example, so the offspring or their kid will carry both genes, actually a mix of both genes. So who is responsible for carrying these traits from the parents to the offspring? It is the DNA. The DNA structure Actually, I'm not talking here chemically. No, I'm talking about the structure of the DNA like in the 3D. Actually, this is called a helix. And this is called a double helix. Here you have two of these helices, and they are like twisted over each other or twisted together. And أقرب شكل in our daily life السبحة. السبحة دي عبارة عن إيه؟ بيدز مرصوصة حوالين بعضها، وتقدر لو انت جيت السبحة دي وقعدت تلفها كده حوالين بعضها هتديك الشكل المشهور جدا بتاع الدي ان ايه اللي هو الدبل هيليكس. يبقى مبدئيا الاستراكشر بتاع الدي ان ايه هو عبارة عن two strands like a strand and the strand another one and they are twisted together to form a double helix structure like this. Okay? Okay. Actually, each of these strands are formed of a series of bases that are arranged very well along each of the strands. The bases, what is the nature of these bases? Chemically speaking, each of these bases actually are nitrogenous bases. We have 4 bases that's very important to know about. They are adenine, guanine, thymine, and finally cytosine. And very important to know their names. Adenine, guanine, thymine, and cytosine. If I tell you they are called nitrogenous base, why are they called nitrogenous? Because they all have the common element, which is the nitrogen. They have nitrogen in their structure. This is why they are called nitrogenous bases. So these bases, for example, the A, which stands for adenine, and then next to it is, for example, thymine, and then, for example, cytosine and guanine. However, they are all forming the 2 strands. Okay? Okay. Actually, here we have the A and T always link up together or join together by 2 hydrogen bonds. And the cytosine and guanine, they bind together by 3 hydrogen bonds. So this is very specific. You cannot say that a guanine will bind with a thymine. No. Actually, they must be complementary to each other, like this and this. And so the thymine will always link up with an adenine by 2 hydrogen bonds, whereas the C and G, the cytosine and the guanine, will join together or will link up together via 3 hydrogen bonds. Okay? Okay, great. So here we have the 2 strands, very long strands of the DNA. Bases, nitrogenous bases, are arranged along the 2 strands, and they twist together in the form of the double helix at the end. Okay, so here again, the A with the T, the C with the G. The A with the T using 2 hydrogen bonds, the C with the G using 3 hydrogen bonds. Okay. If I ask you why DNA is called a polynucleotide molecule, so again, we should not forget that even DNA is a polymer of small molecules. Here I'm talking about this polymer as a polynucleotide. So the building unit is a nucleotide. And what is a nucleotide? Here we go. So here I have the 2 strands of the DNA. For example, I have the nitrogenous base, which is G. Actually, the typical structure of this G or this guanine is a total nucleotide. And what is a nucleotide? Actually, you should know about the nucleotide. The nucleotide is formed from 3 main elements. Like we talked about the amino acid structure, it's very important to know very well the structure of the nucleotide. The nucleotide has 3 main parts. Of course, the nitrogenous base, the one that has the nitrogen, and it has a pentose sugar, which is in our case, it's called a deoxyribose because it lacks one of the O, so it's a deoxyribose, not a typical ribose. And what is the meaning of a pentose sugar? Pentose sugar means it has 5 carbon atoms. Okay, 1, 2, 3, 4, and here we have the 5th carbon atom. Okay, and then finally we have a very important part, which is a phosphate group. So in the exam, you can be asked to draw the structure of a nucleotide. A nucleotide is composed basically of 3 parts. Again, the nitrogenous base, the pentose sugar, which is in our case in the DNA because it's a deoxyribose sugar, and finally we have the phosphate group right here. This is a typical nucleotide. Okay? Okay. What is the importance of the DNA? So I told you that the DNA is responsible for carrying the genetic material and so expressing the traits from the parents to the offspring. However, the sequence of the bases is exactly like a language. You know, we hear a lot today about programming. ولغات البرمجة exactly happens with DNA. It's like a language. The DNA, like the bases of the DNA, for example, ATCG, CGTA, CCGG, and so on. It's like a code, like a shafra. Exactly, typical. I like the word code very much because it's very expressive right here. You can never make any meaning of this huge amounts of bases. Okay, but actually, through a very, very important process that we're going to talk about in detail, inshallah, in its topic, which is called translation. This code is translated into a protein molecule, one that makes very importance in our bodies. Actually, proteins, it's what gives us the shape. It determines how our cells, tissues will develop, and so on. So again, we have the DNA. We have lots of nitrogenous bases along it, A, T, C, and G. They are arranged in every possible way, and it's a very, very, very long molecule. So we have lots and lots of these bases. They make for the code. This code is translated into the proteins, the different proteins of our bodies, which will determine how we develop our tissues and organs and how will we grow. And it also determines our personal characteristics like our eye color, like our hair color, and like our blood group, for example, every single trait. So if you have a blue eye color, this is because of your genetic material, your genes. If you have a brown hair color, it's because of the genetic material, the genes in your DNA. If your blood group is AB or is O, because of the genes that are carried along your DNA. Okay. So here I have a question for you. Forensic scientist, and forensic means like, you know, after a crime has happened and so, الطب الشرعي, in Arabic, they try to find DNA on items of stained clothing. The DNA can be used to identify individual people. Suggest why DNA can be used to identify individual people. You know, in movies and so, لما نلاقي مثلا حد من البوليس داخل and he picks up something like a single hair from the floor or so, and he treats it very delicately, and then what happens? He goes with this hair to a lab and he starts to analyze it for the DNA. Why DNA is used? Why is he doing this? Because he wants to know who is the criminal, right? If he finds the DNA, we'll be able to know who is the criminal. And why DNA is able to be used to identify individual people. This is because every one of us has a totally different DNA. Actually, this difference could come from a very low percentage because the DNA molecule is very large. So all of us are similar in around 98% of our DNA. And the remaining 2% is what makes all the difference in our faces, in our shapes, in our heights, in our weight, in every single trait that we have. So, because individual people will have different and unique DNA, and this DNA will carry different forms and unique forms of the genes coding for a certain trait. I hope this is clear for all of you. Okay. 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 So, I have some question for you. What is the structure of the DNA in a 3D wise? What is the structure?
Two long strands
Yes.
coiled or twisted together in a double helix.
Yes. Excellent. Okay. And chemically speaking, they are formed from. We can call the DNA a what?
Polynucleotide chain.
Yes. And what is a nucleotide formed of?
Three things: phosphate group, pentose sugar, or a deoxyribose,
Yeah.
and a
Right.
nitrogen-containing base or a nitrogenous base.
Excellent. Excellent. Okay. Can you remind me of the names of these bases? Or even
Okay,
the symbols?
there are four: adenine, thymine, guanine, and cytosine, ATCG.
Yes. Excellent. The C and G binds together with or via how many bonds?
C and G, triple hydrogen bonds.
Yes, 3 hydrogen bonds, whereas A and T binds together.
Only double.
Yes, excellent. So here I have like a video for you, a little video, just simply explaining the structure of the DNA.
DNA contains the information that determines inherited characteristics. It has the code for making proteins. DNA is found in the nucleus of eukaryotic cells and in the cytosol of prokaryotes. If we take a closer look at the chromatin inside the nucleus, we can see the structure of the DNA. The DNA has repeating subunits, and those subunits are called monomers, or nucleotides specifically. The nucleotide has three main parts: a phosphate group, a sugar, and a nitrogen base. In DNA, the name of the sugar is deoxyribose, which is part of DNA's name, and there are 4 nitrogen bases in DNA. The bases are thymine, adenine, cytosine, and guanine. 2 of the bases are purines, which have a 2-ring structure, and 2 bases are pyrimidines, which have a 1-ring structure. Adenine and guanine.
That's what part we are concerned with. And so before I leave you, just I have like a flashcard just to recap and refresh what we took today. So first, can you remind me of the reagent used for detecting reducing sugars? It's what?
Benedict's reagent.
Yes, excellent. The positive Biret's test result is?
Violet color or purple.
Yes. Can you remind me of some of the functions of the proteins?
So for growth and repair,
Yes.
production of hemoglobin, production of enzymes, hormones, and so on.
And the one of the immunity?
I'm sorry? And the antibodies.
Yes, excellent. The DNA bases, I'm answering this. A, T, C, and G, adenine, guanine, cytosine, and thymidine. Finally, the ethanol emulsion test is positive with?
A white suspension on the top of the water.
Nope. It's positive with which kinds of foods or food samples?
Fats.
Yes, great, excellent. So anyone that has any question? Any question for me? Okay. Thank you, and I enjoyed my time with you. Thank you so much.
Thank you, Miss. Bye bye.
Bye bye. Thank you.